Hi everyone. So welcome back to another session. So today we are going to have a session on odonates. So introducing everyone to the speaker. So we have Amila from Sri Lanka. So he is basically an expert in odonates, and his primarily work is on their taxonomy and their biogeography. He has been working on Sri Lankan odonates for over a decade and has been widely working with Sri Lankan odonates. He even authored a field guide on odonate of Sri Lanka in 2017 and is very active on iNaturalist with respect to odonate identifications and observations. Over to you, Amila. You're on mute if you're speaking.
Right. Hello.
Yeah. Thank you.
Yes, you can hear me, right?
Yes, clear.
Okay. Uh, right. Thanks for the introduction. And yes, I'll be providing you a brief intro on ordinates, generally what they are, what they do, their behaviors, classification, and how to identify them, and few brief pointers on identifying few key families in our region, in the South Asian region, and maybe little pointers here and there on identifying species and how to record them, how to observe them. If you are uploading in observations to the iNaturalist or any other citizen science platform. So that will be the general flow. I'll first talk about ordinates in general and then move on to the identification and other aspects. If you have any questions regarding any of the slides, any of the information, you can, of course, ask questions at the end. So we can have a discussion time later. Right. Okay. Ordinates, or generally in general terms, dragonflies. Um, Most people generally know them as dragonflies, but of course there are two groups in ordinates which we refer to as dragonflies and damselflies. So there are two different groups, but then in most commonly they are known as dragonflies in general. So these are basically, excuse, right. Dragonflies are insects. They belong to the insect order known as Odonata. That that's why we identify them as odonates or order Odonata. In general, to refer each species, we can refer them as odonates. Commonly, both for dragonflies and damselflies. So they are parts of kingdom Animalia and phylum Arthropoda, then class Insecta, and under class Insecta, the order Odonata. Under order Odonata, there are two suborders, two primary suborders, and there is also a third suborder, which certain people recognize as a different suborder, but certain others don't. Um, Generally, when we refer to odonates, when we talk about dragonflies and damselflies, the two major suborders, Anisoptera and Zygoptera, are the like the primary suborders that we always discuss. Anisozygoptera, the third suborder, is consists of only few living species, only 4 species, if I'm not mistaken, of a single genre. So that's all that is there in Ensozygoptera. So it's not a common suborder that we usually discuss when we talk about odonates, but of course in Asian region, in the Himalayan regions, moving from eastern Himalayas to uh, southern China and then towards Japan, you can see these Ensozygopteran species. Um, Generally, the suborder Anisoptera is what we refer to as okay. No, wait. Where is the laser pointer? That's fine. Anisoptera is what we refer to as dragonflies, and then suborder Zygoptera is what we generally refer to as damselflies. So this image shows a dragonfly, and this second image on the right side shows a damselfly, and the lowest image shows a Anisozygopteran of the third suborder. Right. The main differences between these two groups, Zygopterans and Ansopterans, so the dragonflies and Excuse. Right. The Psychopterans and Anisopteran or the Damselflies or Dragonflies, there are multiple differences. The main difference is actually the shape of their body. The main thing that is generally obvious to anyone when we are observing dragonflies and damselflies is the general body shape. You can see this is a dragonfly, which is like a bulkier insect with more broader body, and they usually in their resting position they usually keep their wings perpendicular to their body, the middle axis of the body. They keep the wings lowered and perpendicular to their body, whereas damselflies are kind of a more slender, long, elongated insect with a very slender abdomen, slender body, and they usually keep their wings along the abdomen. Wings, they close all four wings and then keep them along the abdomen. So that is something that is very obvious when we observe these insects. So that's like the major key difference between the two suborders. But then when you move into their classification, when they are taxonomy, there are multiple other differences that we can observe. One major thing is shown in the pictures below is the position of their eyes on their head. So if you look at the three images under the image of the dragonflies, you can see dragonflies have very large compound eyes, and the compound eyes cover most of their head, right? And on on the top of the head, usually in most species, the eyes meet on top of the head. But when it comes to damselflies, the other groups, Zygopterans, the eyes are located on either side of the head, so they are very well separated from each other. Uh, so they kind of look like a hammerhead shark. Their head, thorax, and head. When you look from above, it looks like a hammer with the eyes, you know, on either side of the head, and then more like a cylindrical head when you look from top view. So the eye position of the eye is something that is that we consider in the classification as as a key character between these two different groups. And also when you look into the details of their wings, the structure and the shape of their wings are also different. Zygopterans in damselflies, usually they have two pairs of wings, forewing and hindwing, and the two pairs are usually more or less similar in their shape and size to each other. But when it comes to dragonflies, usually the hind wing is much broader. It has a much broader base than the forewing. So the wing shape and size is not equal to each other. So that's how they have been named actually. When you look at their names, the Anisoptera and Zygoptera means Anisoptera means in Latin, Anisoptera means different wings, and Zygoptera means similar wings. So that's how the suborders have been originally named. So that these are the key characters that you can use to differentiate damselflies and dragonflies. They are very kind of very obvious characters. Uh, and then moving on to their general morphology, the general structure of their body. Generally, a dragonfly or a damselfly is their structure is very similar to a typical insect, the typical anatomy, typical morphology of an insect. So they have a segmented body. Their body is they're made of multiple segments aligned along their middle axis, and these segments are grouped into three major areas, which we name as the head and the thorax and the abdomen. So the longer abdomen and the middle part thorax, to which the wings and legs are attached to, and the head, which has all the sensory organs and mouth parts. The sensory organs means eyes, antenna, and also their mouth parts. Abdomen is the slender part on the end of the body, which has 10 different segments and the caudal appendages. You can see the caudal anal appendages or the caudal appendages are the kind of a specifically developed structure on the end of the abdomen. And this usually has their reproductive organs and the digestive tract. Thorax is the main main area where where all the flight muscles and legs and all the other appendages are attached to. So the two wings and the three pairs of legs and all the muscle structure that controls the movements of wings and legs are inside the thorax. So this is the general structure, general morphology of a adult dragonfly or a damselfly. Right. Uh, Moving on to more detailed views, when you look at the head, this is the front view of a clubtail dragonfly. When you look at the head from the front view, you can see the two compound eyes, the large compound eye, which consist of multiple thousands and thousands of smaller structures, which we call as omatidia. And apart from these two large compound eyes on the frontal area, you can see three simple eyes as well. These are like very, very simple structures, two, three button-like small structures that you can see only if you look very closely at these insects. So these simple eyes allow them have their near vision, and the compound eyes are very sensible, very sensitive to any movements and all the other sensory vision generally. So the mouth parts are located on the bottom part of the head, and these are consisting of various plates, spines, teeth-like structures, which help them to break down their prey and then uh, eat it. So this is a very structurally very complex structures. When you look at more closer look, when you have a more close look, you can see all these plate-like structures, spines like teeth-like structures, and which consist which makes the mouth parts, and also the omatidia, the smaller uh, units that make their compound eyes. And these very slender, small uh, structures that you can see in front of their compound eyes are their antennas. They have very small antennas, unlike butterflies or moths. They have very small and very simple antennas, just a slender, small needle-like antenna. That's all they have. Nothing structurally developed like the antennas of a moth or even a butterfly. So they both generally leptopterans have more advanced, more structurally developed antennas, but when it comes to odonates, they just have simple, very small needle-like structures. That's all, which is generally not even visible when you look at the dragonfly in general. So you just see it from a normal distance, you don't even recognize the antennas. Right. Then the wings, the dragonfly wing is a membranous structure which is supported by, you can see these vein-like structures, which is kind of hardened areas that support the structure of the wing. And the membrane is, you can see the membrane which is supported by this venation structure. And both wings, the front wing and hind wing, has a kind of a dense area towards the end of the wing, towards the tip of the wing, which we known as the pterostigma, or sometimes known as the wing spot as well. But in technical terms, we call this as pterostigma. Certain species have color patches in their wings, but certain others don't. Generally, most of the species have more like semi-transparent kind of a membrane with no significant color patterns or anything. But certain species have colored veins or colored pterostigmas, differently colored pterostigmas. Generally, pterostigma is either dark brown or black in most species, but certain others have white or more paler pterostigma. And also certain species have especially in the hind wings, certain species have patches in their wing base, which we can, of course, use as identification markers as well. So this is a dragonfly wing that when you look at the forewing and hind wing, you can see the differences that I mentioned earlier. The forewing and hind wing are structurally very different to each other. Hind wing has a much broader base, whereas forewing has a more slender base and kind of a little bit longer than the hind wing as well. So the general structure and shape of the two pairs of wings are different from each other. Right. This is a closer look at the wing base. You can see the structure of the venation and the membrane, and also the color pattern, how it is seen. Right. And other characters, when it comes to their sexual reproductive organs, the male dragonfly, this is a male dragonfly, the one that I showed you earlier. This is a male dragonfly. You can see males are the ones who have these well developed anal appendages or well developed caudal appendages, but females don't have that much structurally developed anus. So when it comes to male reproductive organs, these are the main things that you can see from outside. So one is the anal appendages. Males have a more structurally developed, more three-dimensionally structured anal appendages, and also they also have a organ known as the secondary genitalia, which is located in the ventral half, the bottom half of the uh, second and third abdominal segments. Right. This is when you look at the general morphology. This is where you can see the base of the abdomen is where you can see the secondary genitalia. Right. So, and when it comes to the female anatomy, the female reproductive structures, they have a specifically developed organ to lay their eggs. So they don't have anything similar to a secondary genitalia or even a structurally developed anal appendages. What they have is a some species have kind of a plate-like structure that helps them lay their eggs, or certain others, especially damselflies, have a small ovipositor, like the second picture towards your right, that they insert into the into a plant stem, and they lay their eggs inside. So they have some sort of a organ which allows them to lay eggs towards the end of the abdomen on the ventral side, but males have structure a different organ in the base of the abdomen. So this is something that we always use to differentiate the gender of dragonflies when we see them in the field. Right. Okay. Right. Sexual dimorphism is something that we also observe in dragonflies. In most species, not all, but in most species, this can be seen. Sexual dimorphism is the having distinct morphological, distinct morphs between the two genders, two sexes. The male and female have completely different distinct morphological forms. So this can be observed in multiple species, but I've given uh, two species that you can clearly see the sexual dimorphism. One is the Neurothemis tulia, or the pied parasol, as we call it. Uh, this is the male. The top one is the male. That you can see the black color wing bases, and then the whitish stripe in the middle of the wing, and then the transparent uh, apical part. But when it comes to the female, it's completely different. The color is all the body color, the thorax and abdomen has also a different color, and the wing pattern is completely different from the male. So they show a very distinct sexual dimorphism between the two sexes, males and females. This is also true when it comes to the damselfly that we generally refer to as oriental green wing, or the Neurobasis chinensis, which has very bright, very metallic greenish wings in the male, but the female has completely transparent brownish wings with two cream color patches, one at the pterostigma and one at the nodus, where the wing is bent. So this wing pattern and also the base color of their body is also very different from male and the female. So certain species have this kind of sexual dimorphism, but certain others, like if you are familiar with the common species in the region, if you look at species like Orthotrum sabina or the green skimmer, that doesn't have any sexual dimorphism. The males and females are basically completely similar to each other, other than their sexual reproductive organs that you can see from outside. Other than that, they are generally completely similar. So there are species like that, but then most others have some sort of sexual dimorphism, while certain other species like these examples have very prominent, very distinct, uh, clearly visible, clearly identifiable dimorphism as well. Right. Size of odonates in general, odonates, the body size varies between 15 to 130, 140 millimeters. So that's like 1.5 centimeters to 13, 14 centimeters. So that's like the size range. But then in our region, in Asian region, usually the largest, in South Asian region at least, the largest ones are, I think, close to 11, 12 centimeters. I don't think we have anything that goes to 13. But then we have pretty large species, few very large species in the families like Asianidae. But in terms of wingspan, their wingspan is like from 17 to 190 millimeters. So this is the image shown here, is shows the closest living dragonfly, Nanophyra pygmea, which is known from Southeast Asia to northern Australia. Uh, so this is like the smallest dragonfly species, smallest Ensopteran species, but the smallest themselves, there's no particular species known as the smallest damselfly, but generally if you're familiar with the regional species, the species in genus Agriocnemis, or the wisps, as we call it, the wandering wisp, that group has very small individuals, very small species, but then there are different multiple species within the same size range. So there's no particular species that is known as the smallest, but then there are a group of very small damselflies. These are some of the large ones that you can see. The Tetracanthogyna plagiata, or the giant hawk, the largest dragonfly, is found in the Southeast Asian region. But then the largest damselflies, both these species shown here, the Megalopripus cerulatus and Mesistogaster linearis, both these species are uh, South American species from Amazon region. So I don't in the Asian region, we don't have any very large damselflies. The largest are, I think, the group, probably Orolestes, Singalestes, Megalestes, that group. But yeah, I think that should be the largest dragonfly damselfly group in the Asian region. Right. Life cycle, when it comes to odonate life cycle, odonates show, if you are familiar with general insect life cycles, there are two different strategies that they usually take. There are species that undergoes complete metamorphism, and there are species that undergoes incomplete metamorphism. So odonates are a group of insects that shows incomplete metamorphism. So unlike a lepidopteran or a butterfly or a moth, that has kind of a cocoon stage in between. These insects doesn't have any stage like that. So eggs, larvae, or the nymph, and then directly adults. They don't have a middle intermediate stage that undergoes development within a cocoon. So that development process we call as the incomplete metamorphism. So you have eggs, and then from eggs you get the larvae, which is basically an exclusively aquatic animal, the larval stage, and then you have the aerial or the terrestrial adult. That's the general life cycle. So moving on to different stages of the life cycle, you can see the from eggs, the we get the larva, then larvae lives in the water, and then when the larvae comes out of the water and emerge as an adult, we call that process emergence. So this is kind of a series of photographs of Sri Lankan endemic species known as Microgumphus vijaya. A process, a few pictures of the process of emergence of that particular species. So what happens is they live underwater for many months, and then once they have completed gone through all the larval stages and completed their development, they comes out of the water, and then they grabs onto some structure like a rock or a log or something a hard structure, and then uh, comes out of the exuvia or the these are insects, so they generally have a outer skeleton. They don't have a skeleton like us. They have a exoskeleton, what we call an exoskeleton, which is a outer cover of the body, which gives the structure to the body. So they come out of that exoskeleton, and then uh, by moving liquid that is in their body, they can stretch their abdomen and wings and come to the change their shape to from the larval stage to the adult stage. So this process, the emergence process, takes maybe a couple of hours. It could be completed in 30 minutes in certain times, but certain other times it takes 2 or 3 hours. So this process usually happens in most species. This happens early morning, where they have some uh, protection, because this is like a basically they can't move during this emergence process. They have to complete the emergence, and then they have to dry their wings, and then only they can fly. So the within during the process, they are completely vulnerable to any predators or any other any other factor. So they can't move. They can only crawl a little bit, smaller distances. That's all they can do. So because of that, in most species, this process happens very early in the morning, sometimes even at night. And then in certain other species, you can see them emerging even during the day. Sometimes even around noon, you can see certain species emerging. So this emergence process is uh, this is the process that changes the aquatic larvae uh, to a terrestrial insect. So the process of the uh, morphing into the aerial insect that we usually know as dragonflies is this emergence. So this is something that you can observe if you have a walk along a canal or a suitable aquatic water body uh, in the morning. Especially, you can see, maybe you might be able to see certain species emerging, and once they have uh, become the adult, the within the first few hours they don't have much color, or like they are very how do you put it, they don't have a structural strength. They are very delicate in the first few hours. So if you touch them at this stage during emergence or just after emergence, the state that we call as the tendril stage, if you touch them within during that period, you might actually damage the animal. So if you touch them even by wings, the wings might bend in different awkward angles, and it might not recover. So if you are seeing any emergence, if you are seeing recently emerged insect tendrils, don't ever touch them. So that usually harms them always. So yes. So once this process is completed, once they have emerged completely, and then they stretch their wings and abdomen, and the wings are dried, then they can fly, and they usually fly towards a dense vegetation cover and remain inside for a couple of hours until they gain their full structural strength, and they are able to fly freely. So, yes. Then once emerged as adults, the main evolutionary purpose of their life is to breed, to mate, and then continue the next generation. So the process of mating is something that we generally observe in most dragonflies during these mating processes, because they lay their eggs in water. As I mentioned earlier, their larval stage is completely aquatic. So egg laying happens closer to water, either on water or underwater sometimes, or in plants nearby. So that's where they have their mating activities as well. So if you visit water body during the daytime, you can see many dragonfly is mating or trying to mate or trying to find suitable partners or different stages of this process. The mating process has basically few stages. Um, First, of course, they have to look for a partner and find a partner, and then once they have uh, male has found a suitable female, then the first stage we call as the tandem position, where male uh, male animal catches the female from their prothorax, their head or the prothorax. They use their caudal appendages, the structurally developed caudal appendages, to grab onto the either head or the prothorax of the female, and then that position, the first position, is known as tandem position. During this process, we see most animals during the mating process. We see most animals during tandem stage. They can fly while being in tandem. So this is something that we very frequently observe. Uh, in this position, always male is the one on top and the female is the one on the bottom. So they in dragonflies, the anisopterans, male always catch the female using the back of their head. There's a groove-like structure in the back of their head. That's what they use to catch the female. But in damselflies, this happened. They use the female's prothorax or the front part of their thorax. So they have kind of a slight differences in their mating process, but then generally they follow the same behavioral pattern. Um, right, and then once in tandem, then they forms a position known as the mating wheel, or the uh, sometimes this is known as a wheel position, or sometimes it's also known as a heart position, because of this kind of a very little bit heart-like shape that they form when they comes into the mating position between using the abdomen. So because of this, this is sometimes known as the heart position, sometimes known as the mating wheel or the wheel position. So this is the stage that they actually do the mating and exchange their do the sperm transfers. So dragonflies and themselves in odonates, the male develops their sperm in their reproductive organ, which is located at the end of the abdomen, and then they transfer this sperm to their secondary genitalia, which is in the base of the abdomen. That's where the female attach its mating reproductive uh, organ to the secondary genitalia, and then the sperm transfer happens. So that's how they do mating. That's why they form this kind of a mating wheel, unlike most other insects that they usually touch the tip of their abdomen while mating. But damselflies and dragonflies have a different mating position. The female attaches its abdomen tip to the base of the male's abdomen, and then sperm transfer happens. So during this position, they they can stay sometimes in certain mating behavior, certain mating rounds. They can stay for many hours in this position, but generally the mating happens within a very small period. But what happens is the male dragonfly or the male damselfly is able to, once this connection is made, once the female reproductive organ is attached to the male secondary genitalia, the male is able to, if the female has been uh, mated before, if it already has sperm within its reproductive tract, the male is able to remove that sperm and uh, release it, replace it with his own sperm. So this process, the removal of sperm and then replacing it with its own sperm, sometimes takes time. So because of that, so in certain mating certain mating occasions, that can go up to uh, maybe 1 or 2 hours even. So this is something that odonates have developed behavioral uh, behavioral certain behaviors that ordinate mates have developed to increase their fitness, to increase the reproductive fitness of the male. So it's kind of different from many other insects. This is something very unique to odonates. This sperm transfer using the mechanical removal of sperm and then replacing with its own sperm. So this has been studied for long, ever since the odontology has been developed. This behavior is something, the sperm competition and these different mechanisms that they use to remove the sperm and replace it with their own sperm is something that has been studied very thoroughly. So yes, so once that is also complete, once the mating is also complete, then the female moves on to uh, laying their eggs. So that part could happen in different ways, depending on different species. Right. So I'll give you a few examples. Right. Yeah. So this is like the the male usually uses two strategies when they are finding females. They have they either find a suitable ovipositing habitat, a suitable habitat to lay eggs, and then form a territory and then remain there until the suitable female comes. Or certain other species patrol the entire area until they come across a suitable female to mate and lay eggs. So there are two different strategies: sit and wait kind of a strategy, and then actively go look for a suitable female and then uh, continue with mating. So there are two different strategies that are used by the different species. Certain species are more like a patrolling kind of species, like these large dragonflies. Most of the large dragonflies, like Aeshnids, darners, some clubtail species, they use that strategy. Cruisers, especially species like Macromia, they use this. Whereas most of the libellulidae species, or most of the damselfly species generally use the uh, territory forming behavior. So there are two different strategies. And during this process, especially when it comes to territorial species that forms a territory, uh, you can see certain territorial behaviors between males. Yeah, once a male dragonfly has formed a territory, if a male of the same species enters that territory, they usually fly out and interact with this second male and try to chase it away by having kind of a not really aggressive, but kind of a conflict, kind of a mating combat. This can be seen in different damselflies, but mostly in damselflies. But this is specifically seen in species like libellagos, or euphea, or even species like sometimes vestalis. So these species show this particular pattern of kind of a dance-like uh, 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 dance-like behavior, that they interact with the other male and maneuver themselves in a rhythmic pattern and try to figure out who is the strongest, who has the uh, who is better fit for the territory. So I don't know how they actually kind of compare each other, but then probably the symmetry of the flight, or the something must be the triggering factor to decide which individual actually owns the territory, and uh, which is the second the which leaves the territory. So that's, I don't know how they actually uh, decide that, but they fly in a very rhythmic pattern, very kind of a dance-like pattern, and then that's their combat ritual that they perform. So this can be seen in many species, not all, but certain species within the Asian region. So that's some kind of a strategy that they use. Certain other species, like large dragonflies, sometimes aggressively attack each other. They fly into the other, fly towards the other individual and kind of directly physically attack it. Uh, uh, so that's another behavior that certain other species show. But in some manner they decide who owns the territory and who leaves the territory. So the best, so more stronger, more suitable individual usually uh, retain the territory. So yes, and then this is like uh, other damselflies and dragonflies mating wheel position. You can see how the female individual, the one on the bottom, how it has bent its abdomen and um, connect with the secondary genitalia of the male. And here also you can see the same. This is a species called known as Seriagrion cenorubellum or painted waxtail in our terminology, but this is a species known as Orthotrum prunosum or the pink skimmer. So in both these suborders, the same position can be seen, but with certain slight differences. When you look at when you have a closer look at the where the male has grasped the female, you can see the two differences where the dragonfly has used the uh, grasp the female using its head, whereas the damselfly male has used the prothorax of the female. So you can see that two differences very clearly. And this is the mating. This is how the female genital opening is connected to the secondary genitalia of the male, and this forms kind of a lock and key kind of a mechanism where each species have a structurally developed secondary genitalia. Each male of each species have a structurally developed secondary genitalia that can perfectly fit into the uh, reproductive tract of the female. So between species, the structure of this structure of the uh, structures in the secondary genitalia of the male changes from species to species. So this is another character that is used in the taxonomy in identifying species, classifying species. But when it comes to very detailed level of taxonomy, not just field observations, we don't usually look at secondary genitalia of the male when you identify them in the field. But when you do the taxonomic work in the laboratory, you usually use these kind of characters. Right. Moving on to oil position or the egg laying process. Different species, different groups have different egg laying strategy. What happens generally is dragonflies, in general, they usually lay their eggs in water, whereas damselflies usually, in most cases, they lay their eggs within a plant matter, within a plant tissue. So this is what the general dragonflies do. Most of the skimmers and even gomphids, what they do is they either fly above water and then the female releases its egg while flying, or they sometimes tap the surface of water using their abdomen tip and then release their eggs. Certain other species do do the same, but then they do it only between aquatic plants. Only where you have aquatic plants on the water surface, they tap their abdomen tip and on the water surface and then release eggs. So different species have different strategies. Certain species completely lay their eggs in open water where you don't have they don't need any aquatic plants to be there. But certain others lay their eggs where only where aquatic plants are available. And some do this while flying, some do this while flying, but then they tap their abdomen tip to release their eggs. So different behaviors, different strategies based on different species. So this is an example like that, photograph taken between egg laying process. You can see eggs, clutch of eggs in the abdomen tip of this female. This is a species known as Wall's purple tail or Heliogomphus walli, which is again an endemic species to Sri Lanka. So this was taken mid oil position somewhere, yeah, in a rainforest stream during the like midday time. Right. Different strategies. Certain species have different strategies. One on your left is a Dana, which is the species is Annex immaculifrons or the fiery emperor, and the one on your right is a smaller, very small dragonfly of the family Libellulidae or skimmer, which is known as Tetrathemis yerberi or yerberi itself. Again, an endemic species of Sri Lanka. But Tetrathemis genus is found at other places in Asia as well. You have Tetrathemis platyptera from South Asia, South India to Southeast Asia, and there are other species in African region as well. So the different strategies that dragonflies use. One, the darners, the species in family Aeshnidae, they lay their eggs w- just like damselflies. They lay their eggs inside plant tissues. So they sometimes they lay on the surface of water and then dip their abdomen tip underwater and then release the eggs into a log or a root or leaf litter or some kind of a plant tissue. So this is something that you this is not something that we generally observe all the time. This uh, I have seen this egg laying process of a darner very few times, but that's how they lay their eggs in general. Uh, Other species, the Tetrathemis or the Yaburi cells, that also has a very different strategy. They are the only ones that lay eggs open to the atmosphere. They attach their eggs to a structure above the water, usually a hanging root, or a small branch, or a tip of a leaf, something like that. They attach their eggs there, and it's basically open to the atmosphere, open to the air until the egg hatches and the larvae drops to the water. So this is the only species that do use that kind of a strategy, that lays eggs open to the atmosphere in our region. This particular genus, Tetrathemis. So these are like very different strategies used by different groups of dragonflies. Damselflies, again, as I mentioned earlier, they usually insert their eggs into a plant tissue. While doing this, certain species can go completely underwater to do that, and then insert their eggs into a mosses or underwater roots or any other or a fallen log or stick or something, any plant matter. Uh, Certain species can completely submerge themselves for a period for maybe 10-15 minutes, while certain others don't completely submerge, but they only submerge their abdomen tip and then lay the egg. So this is again a process that we can see among many different groups. Generally, if you look at the ovipositing behavior, egg laying behaviors of euphias or the gossamer wings, what we call as gossamer wings. Euphia in South India, you have multiple Euphia species in along the Western Ghats, Euphia fraseri, Euphia, all these different species. When we only have one Euphia species, what they usually completely go underwater, and they can stay underwater for a very long period of time. Like I've seen them being underwater for like 30 minutes, and but there's a single record from, if I'm not mistaken, there's a record from Southeast Asia where Euphia has been observed to lay eggs for almost an hour, for, if I'm not mistaken, 58 minutes or something, that they have submerged themselves and continue to lay eggs. So this is something that we always see in Euphia species. But certain other species like Elatenura, uh, threadtails, or Pseudacreon, you can observe this underwater posting behavior. Also in the species known as marsh dancer or the Onychagia atrosina, they also do that. So there are multiple species that perform this underwater ovipositing behavior, but certain species are well known for that. Other species generally lay their eggs in plant tissues above water. It could be on the surface of water, it could be above water, a grass or a reed coming out of water, any material that they can, any soft plant material that they can penetrate and lay the eggs inside. So that's the only requirement there. Um, Males usually, while the female is laying eggs, usually in most species, not all, in most species, males guard the ovipositing female because of the reason that I mentioned earlier. Because if a different male comes and grabs the female, it can remove the sperm that the first male has uh, placed in the reproductive tract of the female and then replace it with his own sperm. Because of this risk, usually the male guards the female until the oviposition is complete to ensure that the sperm that it has released onto the body of the female is used for egg laying process. So otherwise, it will be a waste of energy on its behalf. So it won't give it won't secure it won't create uh, any offspring from its own sperm. So that this because of this, the evolution has given them this behavior because evolutionary all these species what they the main purpose of their existence is to continue the uh, continue the generation and to make sure that they have as many offsprings as possible. So that's the evolutionary purpose of all these animals. Because of this, they have this guarding behavior. We call it guarding behavior. They certain species do the guarding while flying above the female. Certain others do it while perching closer to the ovipositing female. Certain others also remain in the tandem position, keep grabbing onto the female while egg laying is complete, like this one, this photograph. So yeah, this happens in many different ways, but still the uh, purpose is the same. Right. Other behaviors that you can see from odonates, there are various different behaviors. How they catch prey, how they fly, and uh, the behaviors have been studied for a long time, especially their flight abilities and things like that. But something that we, of course, observe when we go onto the field and observe this is this obelisking behavior. We see species like this, individuals like this, completely they have completely lifted their abdomen and remain in the vertical position. This is something that we usually recognize as a different behavior. So this obelisking behavior is has been developed as a thermoregulatory behavior. Uh, Dragonflies usually do this only during midday where heat is very high. They try to prevent the surface area that is exposed to the direct sunlight and minimize the minimize the heat the exposure to heat. So to prevent desiccation. So this behavior is a particular behavior that is mostly seen in dragonflies. Damselflies don't do obelisking, but damselflies also shows other thermoregulatory behavior, like they sometimes go on the underside of a plant material and then remain vertically hanging to prevent exposure to sunlight. Certain species do that. So there are other thermoregulatory behavior as well, but this is something that we generally see. Other interesting behaviors, other interesting facts about these animals are they are generally most dragonflies and damselflies are highly seasonal in their within their life cycles. So the adult stage that we generally refer to as dragonflies and damselflies, when we generally talk about dragonflies and damselflies, we are not referring to their larval stage much. We are talking about the adult. So the adult stage is usually very seasonal, in especially in species that are adapted to forest settings, adapted to forest habitats, uh, mountain habitats, small streams, and inhabiting these more humid environments are usually very seasonal. The charts below shows these charts have been taken from a publication on Sri Lankan odonates. So this generally shows how the number of individuals, number of observations have varied through between during the different months. So you can see certain this particular individual, again an endemic species to Sri Lanka, is generally seen from April, May, June, July, that period where we get the southwestern monsoon. the monsoon rains are there, so more water, more streams are filled with water. You have more oil posting habitats. So they are only seen on wings during that period. Whereas in certain months like towards December, January, February, you don't really see them around. You don't even see a single individual of certain species during these dry months. Whereas more generally species like more species that usually are inhabiting open wetlands, open habitats, not forest dwelling species. They these kind of generalist species they can be observed throughout the year, certain species, but then again not everyone. So there's a clear peak abundance in the rainy season, in just after at the beginning of the rain season and the during the rain season there's a clear peak of abundance among many dragonfly and damselfly species. So that's something that we should always keep in mind. Even the richest area, even the area with the highest number of species would have nothing on certain months. uh, I have been to certain areas like in when it comes to again Sri Lankan context. There's a rainforest area that is famously known as the Singharaja rainforest, which is like a UNESCO World Heritage site where we have like the highest number of dragonfly species, dragonfly and damselfly species in within the country. But if you visit the area in January, February, I have visited January within in January, February, and have not seen a single individual of any species. So that this kind of seasonal patterns are there in dragonflies, odonates in general. So that's something that we should always keep in mind. And when we are doing our visits, when we are doing our walks, nature walks, if you are looking for dragonflies, we should always uh, plan it at the right season. Um, yes. Also, another behavior that is known among dragonflies is their migratory patterns. Even though they don't migrate like birds do, birds, of course, are well known for their annual migrations, seasonal migrations, uh, north-south migration, and certain species or east-west migrations are also there. So these kind of migratory patterns have been very well documented and well known among birds. But dragonflies don't migrate like that. But then still certain species show migratory patterns like these, the species shown here, the uh, wandering glider, Pantala flavescens, or sometimes also known as globe skimmer. This is a well-known species for their migratory patterns, and it is a species that shows the longest insect migration in the world. They migrate from South Asia. They fly from South Asia towards Eastern Africa, along Maldivian islands. So that migrate that flight from South Asia to Eastern Africa is the longest known flight among insects. Longest known flight of a single insect. The whatever the individual that leaves India enters Africa. India and Sri Lanka enters Africa. So because there are no large no large uh, islands, freshwater sources for them to breathe. So whatever that leaves the uh, western Indian and Sri Lankan coast enters Africa, and then they migrate within Africa, and then come back. The next generation comes back to South Asian region as well. So the entire cycle consists of multiple generations, multiple individuals, but then this direct flight from South Asia to Africa is the longest known distance of migration for a single insect. The other very well-known example for insect migration is the monarch butterfly of Americas. But then monarchs also the entire migratory cycle consists of multiple individuals. They certain individuals fly for certain distance and then they breed, and then the next generation of adults fly continue the migration. So that's how the uh, entire cycle is completed. Uh, yeah, so there are several other species that are known for their migratory patterns in other regions apart from Pantala. There are certain species of darners that are known for migration, and also even Certain others are also known for their long distance dispersal flights. They are not migratory patterns. We call migration when it's like a cyclic movement. You whatever that flies, and then it use the same cycle, same path every year. There's a seasonal pattern to it, but they are the dispersal flights, what we call when the new adults are emerged, they uh, fly towards a different region, different area to find more breeding habitats. That dispersal movement, some species, is known as very long distance dispersal movements among certain different dragonflies. Because of that, you sometimes get very odd records. Uh, African species coming into Asian region, very rarely vagrant movements of species. That kind of observations are also there because of this long distance flying ability. Yes, so this is the general scenario, general behavior patterns and general morphology of odonates in general, both dragonflies and damselflies, and that's like the major part of the my presentation, to give you a general idea on what they do, how they do it, what behaviors are there, what evolutionary adaptations are there, that kind of information. And moving towards the identification component, uh, the first step, if you are completely new to this subject, completely new to identifying dragon odonates in general, the first step is to learn how to identify dragonflies and damselflies. So that's a start. That's basically, if you have 100 species, 50 dragonflies, 50 damselflies. If you know how to separate them, so you bring it down to half of that potential species. So that's a start. Always you can start with that. And then another important factor is to identify sex, the sex of the individual that we are looking at. Because most of the times, the field keys, the identification keys that we have developed are usually suitable for males. So because males have more colorful morphology, and also they also have, because of the reasons that I've discussed earlier, they also have more structurally developed, more structurally uh, distinct organs like the caudal appendages and secondary genitalia, which is absent among females. You have certain structural characters among females, but then it's more prominent among male individuals. So the keys that we usually use, the gender identification keys, are usually developed for male species, male individuals. Um, so it's always good to identify the sex of the individual that we are looking at as well. And then if you know how to identify different families, and then within families you have different genus, so then identification process can follow along that line. And you can, if you know how to use a key identification key or a field guide, you can easily follow the process and uh, identify the individual that you are looking at. uh, I would discuss few families in the region. I generally use what is available in Sri Lanka, what is present in Sri Lanka, but these are also the same families that is known from the South Asian region, even in the Southeast Asian regions. These are like the uh, major families that you can observe. So I'll give brief intro to each family, main key, kind of a main one or two key points on how to differentiate them and how to identify them. So first family, a well-known family among Asian damselflies, is the family Chlorosiphidae, which we generally refer to as jewels. Or of this family, the one of the most common species are the genera Libellagos, uh, Libellugo, and there are other genera that is present in Asian region, South Asian region, like in Western Ghats you have Calocipha, in uh, Himalayas you have other Aristocypha and other species. So Heliocypha also there, Rhinocypha is there. So there are few popular genera. So the main character among these damselflies are they are very small. They are less than an inch, some of the individuals, and they always have longer wings than their abdomens. So that's a character that is only seen among chlorosiphids in this region. Most of the other damselflies doesn't have this. Many species have much longer abdomens than their wings. So this is a key character for this family. They have shorter abdomens than their wings, and they are very small. You will never see a large chlorocyphid. All chlorocyphid species are very small. Maximum maybe a 1 inch or maybe slightly larger than that certain species, but in whatever the species that we get in our region are smaller than that. So that's one group. They have different colors, different individuals, different gender have different colors. So colors and everything is different. But these structure characters, the proportion between wings and abdomen and the gender small size is uh, are key characters that you can use to identify these. Then you have another group of damselflies known as broad-winged damselflies. There are two major groups among these general broad-winged species. One is family Euphaidae, and also the other is family Caloptrigidae, the one on your left, and Euphaidae is the one on your right. So Caloptrigidae species have comparatively very long legs. If you look at this picture, you can see they have very long legs compared to their body size. And also they usually have metallic greenish, bluish kind of body, uh, whereas certain species have more colored reflective wings like this Neurobasis species, but others have transparent or sometimes species with just only the wing tip uh, being black or faintly brownish, something like that. So the general factor is their wing shape, as mentioned by their name, are very broad. They have very broad wings compared to other damselflies, and also they have very long legs, comparatively very long legs. Those characters can be used to identify the family Caloptelgidae, uh, of which we have multiple species in our region, and also the family Euphaidae, the second family. They also have very broad wings, and you can see in both species, both these families, you can see the forewing is slightly longer than the hind wing. So that's again another uh, character that you can use to identify them from the other animals. And Euphaidae also have very particular shape in their wing, much broader middle section and very broad wing in general. And these species have different colors. This is the only species that is found in Sri Lanka, the Euphia splendens, which is black marked in slight yellow color in younger individuals. But then whatever you have in whatever Euphia species that is present in Western Ghats are completely different to this. They are red and black in color with uh, partially pigmented wings. They don't have completely pigmented species. So whereas when you move towards Southeast Asia, you have multiple other morphotypes, dark species, blue color species, partially wing pigmented species, completely pigmented species. There are multiple other different uh, types. So these two groups are again prominent Asian damselfly groups that you can see during your walks generally, especially in forested areas. Right. Another main group that is like that has a worldwide distribution is the family Lestidae or the spread wings. As mentioned, as explained by its name, it has enduring resting position. Usually, most species keep their wings spread open, so their wings are partially open uh, during the resting position, unlike in other damselfly species where they usually keep their wings along their abdomen. There's a there's a group of lestids that don't do this. They keep their wings similar to other damselflies along the abdomen, but most of the Lestidae species have this character. So th- they are not very numerous, but they have multiple species in South Asian region, and there are certain genera that have worldwide distribution. So they are like very not very common. You don't see them in thousands, but then they are like everywhere. You can in any habitat you can see resting. Uh, Another major group that you don't really see in large numbers is again the shadow damsels or the forest damsels that we call as family, known as family Platystichtidae. These are generally dark colored insects, dark colored damselfly species. All these species have a blue abdomen tip, and certain species may have dark wingtips, but that's only with only between a few species. Only few species have this dark black color wingtip. But generally, all species are dark with a light bluish, pale bluish abdomen tip. The actual character to differentiate them is in their wing pattern, wing shape. But in general sense, you can, if you see a dark black or dark brown, very not very colorful kind of a damselfly with blue color abdomen tip, it could be a Platystichtidae species. Um, Western Ghats has multiple species of genus Protosticta and also an endemic genus called Indosticta, and Sri Lanka has an completely endemic lineage of multiple Platystichtids. So it's a very diverse group when it comes to the Asian region. It's very diverse in the Southeast Asia as well. So there are hundreds of different species, and they have very minor characters to separate these species. You usually have to look at the details of anal appendages or details of prothorax or closer. Uh, you have to closely examine it, and then only you can identify certain species. So they are not very colorful. They don't have different color patterns and things like that. So you have to rely on the details. The actual detail that the key character that separates them from other themselves is this particular wing shape. If you look at this Platystichtidae wing, there's a kind of a bent. Uh, the wing is kind of bent in the ventral side of the tip, so that is the character that they use to taxonomically separate this from other damselfly groups. Right. Platycnemidae or the threadtails or feather legs are another group of common damselflies. Um, they have multiple species again, and they're generally not all species, but generally certain species have kind of uh, flattened flattened legs, the laterally flattened legs, which gives the name the feather leg. If you look at this species on your right, the Pseudocopera ciliata, uh, you can see this kind of more feather shaped leg, which is different from most damselflies where you have more cylindrical slender legs. So this has a very different shape to its leg. This is not visible in all species of Platyctidae, but then many species have this. Right. Bluets or family Synaglionidae, again, this is like a mixed bag. There are multiple different species in this family, very large number of species, and they have all sort of colors, patterns, and everything. So there's no particular uh, similar morphological character. But most species, not all, but most species have what we call as postacular spots, or the small bluish or whitish pale color spot on top of its head, uh, above the eyes. So this is a character that you can see in many Synaglionids, many bluets, but not all. Certain others have different other characters. So this could be this could the shape and structure of the shape of the spot could differ. Certain species have these slender arrowhead-like marks, while certain others have more blotch-like marks. So anyway, they have these postacular spots among many species. Right. Moving on to dragonflies, one of the largest, a group of very large dragonflies, darners. They have most species are close to 10 centimeters, 10 centimeters in length. So these are very bulky, very heavy, large insects, and they have very broad wingspans as well, long wingspans as well. The key character among this group is the when Now these are dragonflies. I mentioned that dragonflies have compound eyes that meet on top of their head, and in danas, the eyes are they touch each other along the almost the entire length of the eye from the front to the back of the head. The entire length of the eye uh, is touching the other eye. So that long contact between the two eyes is a key character that we usually use to differentiate Danas or Asianids from other dragonfly groups. On the other hand, when it comes to the family called Gomphidae or the clubtails, you do have a gap between their eyes, but not like damselflies, not like what was discussed in damselflies, but unlike most dragonflies, they have a clear gap between the two compound eyes. So that's again another character that we use to identify the gomphids or the clubtails. And gomphids in this family is generally dark with pale yellowish or pale greenish markings. So that's like the universal color pattern that they have all over the world. Generally, they are black with some sort of yellow or green markings, or sometimes they can be more greenish or yellowish with small black uh, stripes. So they can be more yellow or more green, but it's always black and yellow or black and green in most species. Right. Cruisers or family Macromidae is another common group of dragonflies that we can see. They do have eyes touching, like only part of their eyes touch each other, but uh, they are also medium-sized dragonflies. The main character is they have, unlike most other groups that was discussed earlier among dragonflies, they have metallic colored bodies, and their eyes are also usually they have kind of a metallic reflection, metallic blue or metallic greenish reflection. So and also their body also have, if you look at this picture, you can see kind of a metallic bluish reflection is there. So that's a general field character that you can of course always use to identify Macromidae family. Or when it comes to South Peninsula India and Southeast Asia, there are other multiple other families that uh, somewhat look like this as well. Species like Idionyx and Macromedia also have this kind of metallic uh, color to them. But the entire group of these macromid uh, dragonflies have these metallic color patterns. Right. And lastly, the libellulid, or the most generally common species of dragonflies, forms into this group, the skimmers. They are generally very open wetland habitat species, marshlands, water bodies, tanks, are the habitat ponds are the habitats that are generally used by most skimmers, whereas few species are forest specific, but these are very generalist kind of dragonflies among worldwide. I mean, they are very open habitat, uh, prefer open places. Um, right. So they also have that they are head. If you look at the compound eyes, only part of their eyes meet in the middle, just like macromids. So they both have these characters, whereas only the danas have this more elongated contact between the eyes. Right. So these are some general groups of dragonflies, and other factors to consider when you are identifying dragonflies and damselflies in the field is there are certain individuals that have. Uh, I mentioned that the identifying the sex is important, but there are certain individuals that have different color patterns to their normal sex. For an example, these andromorphs and gynomorphs, what we call as the this if you look at these pictures, you can see this is the species known as Eurothemis signata, scarlet basker. This is the top left is a male individual, top bottom left is a female individual, whereas the other image shows a female with slightly with a color pattern which is more similar to the male. That kind of females having male slightly more male color forms, and very rarely males having female color forms is also a phenomena that you can see among dragonflies. So it is a fact, it is a phenomena that you have to be careful when you are making field identification, especially even though certain species have male coloration, it might not actually be a male. This is also true when it comes to certain other damselflies like these blue tails, common blue tail, Ischnura senegalensis. You have different forms of females. Some females, like this one on left, some females are more similar to the male in color patterns, whereas certain other females are completely different to, generally different to male with different distinct colors. Uh, certain We are not 100% sure of this, but there's a the general understanding is that younger individuals, younger females are the ones with orangish color patterns, and when they develop, when they get matured, they become greenish. That's just an observation. I've never tested it actually. So in any case, they have multiple different color forms among female dragonflies, damselflies. Also, when it comes to males, usually during their early periods, the immature males are similar to the male in color. The purpose of this is that it's not having female colors. It's having more less colorful patterns, because why, if you are colorful, if you have brighter reds and blues and all these bright colors, you are easily visible to predators. So females and younger individuals usually have uh, more dull colors, so it gives more better camouflage. And once they develop, when they are sexually matured, when they are entering the uh, uh, reproductive phase, they develop colors for their attracting males, attracting females, and competing with other males. So that's a secondary thing. Because of this, in most species, the younger individuals, younger males are uh, more dull colored, more similar to the female of the same species. So that's another factor that comes into the play, that people easily make mistakes between females and younger males among certain species. So this is the species known as Prithemys aurora. It is the mature male, and this is the immature male. It's completely different in color. You can see the anal appendages; they are structurally same, and the shape of the body is also different from the female. Right. Another example of age-dependent color. This is a immature specimen of Indothemis limbata, and this is a mature specimen of Indothemis limbata. You have certain color patterns that is more visible in younger individuals, but they become more, uh, less obvious in matured individuals. So that's another uh, observation. And also the last factor is the prunescence, or the powder-like dust that develops on their body in certain species. This is a good example for that. Especially among uh, agriocnemists, wandering wisps and other wisps, this can be seen. There are individuals which is completely structurally similar to others, but they are completely white in appearance because of this dust-like particle that they develop on their skin, which we call as prunescence. So these individuals are more whitish, more grayish or light bluish than the other individuals, but they are the same species. Structurally, they are the same species. So this fluorescence is something that you can see in certain different groups of dragonflies and damselflies. So that's something that we have to pay attention when we are making identification as well. So if you see a pronouns individual, you have to make sure that you look at the structural characters as well. Right. General sense, if you want to identify an individual of dragonflies or damselflies, look for colors and color patterns. In most species, I would say, at least in Sri Lankan scenario, definitely up close to 80% of the species you can easily identify if you are familiar with the color patterns, colors and color patterns. And also size and shape is also very important, because different if you are familiar with the birding concept, we have something like as a concept called as GIS, GISS, general impression of size and shape, which we always use when we make identification. That is true for these animals as well. That's true for dragonflies as well. The general impression on their shape and size is a key character that you can uh, that help in the identification game. And also, certain species have wing patterns that is visible during flight that can be used as identification markers. And if you are going to more detailed identification, especially in certain groups like clubtails, Platystichtidae, shadow damsels, and other Protosticta, Indosticta, that group, you have to look at anal appendages of males in most cases. And certain cases, secondary genitalia uh, also comes into the picture. Species like Orthotrum, they are very similar in certain species are very similar to each other, and you have to look at secondary genitalia. So those are few things that comes into the uh, identification process. And also when it comes to females, in most cases females, they of damselflies, they have structurally different prothoraxes that can be used in the identification process. Because I mentioned earlier that the male damselfly usually hold on to the female using its prothorax. So the prothorax of the female has developed structurally to match the anal appendages of that particular species, particular male. So both male and female have structural adaptations that allow them a perfect match between only between that species. So because of this reason, certain individuals, certain species have uh, structurally develop characters in their prothorax of the females that can and have been used as taxonomic characters in the identification process. So these are things that you can consider while you are making identifications. And if you are as a citizen scientist, when you want to make observations and upload them into citizen science platforms like INET and get identification help, uh, make sure that the only key thing is make sure that you get a clear picture. Ideally, for many dragonflies, not all. I mentioned certain groups you need detailed characters. If we can't see the detailed characters, you can't uh, make a confident identification. You can make a guesswork. You can come close, but then you might never be able to confirm the ID until you see certain structural characters. But then in most species, if you get a good what we call a dorsolateral shot, a shot from a side, a little bit towards the top angle, so you can see the top dorsal side of the animal as well as the lateral side of the animal. If you have image like that, in most cases you can make a uh, pretty decent identification uh, on most of the species. So an image like this, from a dorsolateral angle with no disturbances or other interfering objects or anything, or light patches and things like that. If you can get a clear picture within from a decent distance, it doesn't have to be a macro shot, up close. Even 1 or 2 feet away, you can make a decent identification for most species. So try to get a clear picture from a dorsolateral angle, and that would work for most of the damselflies and most of the dragonflies. But also keeps in mind certain species cannot be identified from that only. You need to see detailed characters. So without observing detailed characters, without observing the anal appendages or the in certain cases wing venation or certain prothorax structure or something, no one can actually confidently make identification. So that we have to accept that as well. That's a limitation that is out there. Even us, even been even our experience of working with these animals for over 10 years, we still can't make a positive ID on certain images because you don't see enough characters. So that's a reality that you have to accept. So make sure that you try to get a good angle and a good this interference less good decent image. Then that would positively ensure that you get a decent identification in citizen science platforms like INET. So that's about it. And also, if you're making any field walks, nature walks, uh, in hope of observing ordinates, make sure that visit wetlands. That's where you can find most of them, and not just along the water body, even outside, even slightly outside where you have dense vegetation. You can find most of the females are usually found among these vegetation, not exactly on the water. So pay attention to that as well. And you can see them easily throughout the day when the sun is out there. So they need sunlight. So early morning, like 6 a.m., you won't see anything. 7 a.m., you won't see anything. After maybe 7:38, then you start seeing certain individuals coming into the water body. So there should be enough sunlight that falls on the ground and water. So it has to uh, the temperature must be within a certain limit that they can actively uh, come and do their engage in their behaviors. So because their body temperature depends on the environment temperature. So early morning, usually they fly above, stay on the trees and expose themselves to early sunlight. And then once their body is heated up, they come to the water body to breathe and you know feed, breathe, and other activities. So only after about 8:30 you can see many dragonflies flying around, but before that you won't see many. So early morning, like it's not like seeing birds. Early morning walks won't provide you much when you're looking at dragonflies. During daytime until about 4:35, you can make good observations. Certain species like all the cruisers, macromids, certain clubtails are active usually around dusk. So that's another thing. So there are certain species that are more active in the dusk, uh, after 5:30, 6 at that time, or during an overcast day, they might be active during daytime as well. So there are different species have different patterns, but when you are in the field for a while, for some time, you recognize these patterns. Okay, so these are the things that I wanted to discuss with you. I think we've taken 1.5 hours target time. If you have any questions, of course, you can ask them. If not, we can discuss through other communication channels as well. Thank you very much.
For the great session, sir. There are some questions in the chat. If you want me to read it out, I can read out to you.
Yes, please. Go ahead.
Yeah. So, sir, Seth asked, do dragonflies play a role as bioindicators of ecosystem health, similar to butterflies? Kindly guide.
Yes, I would say they are even better than butterflies, especially when it comes to freshwater habitats, because different species have different tolerances for because they have exclusively aquatic larvae. So they lay their eggs in aquatic habitats. Their larvae is completely living in that aquatic habitat. So the larvae generally has some sort of a tolerance level for the aquatic quality. Because of this, there are certain species that can tolerate contaminated water, like the Brachythermis contaminata, which is found even in urban areas, even city ditches and gullies and everywhere. But most other species cannot tolerate that. Whereas certain species can tolerate estuarine water, but many others cannot. So certain species are completely sensitive to most of these contaminants, so they can only tolerate reasonably quality water in forest streams, just like that as well, like that. So they are a very good indicator for aquatic environment health. And they also have been certain indexes have also been developed using dragonflies. Not in our region. I have never seen a good proper quality index in this region, but there's a South African uh, aquatic habitat quality index that has used dragonflies. There's a whole separate publication on that. So they have been certain indexes have also been developed, so they can be used as a good environment quality indicator. I would say they are even definitely better than butterflies. They are more sensitive to aquatic habitats. So yes.
Yes, thank you, sir. That makes it a lot clear. The second question is how to distinguish between juvenile versus adult odonates for distinguishing male and females.
Juveniles versus adult, once they have emerged as an adult, you don't see any structural differences between juvenile and adult. Like immature adults and mature adults, you won't see any structural differences. The only difference is in their colors usually. As I mentioned earlier, male immatures have more dark colors, while mature males have more brighter colors. So that kind of differences are there. Also, the all individuals are you can see the wings are worn. You can see wings have been damaged. So there are other physical uh, things that you can observe, damaged wings. Like that. And prunescence is also something that usually develops with age. So all the individuals have more prunescence, like I mentioned, this whitish, bluish, dust-like pattern. Uh, that is also there. But once you have emerged as an adult dragonfly, there are no clear, significant, like structural things that you can see. Only the color patterns are different.
Hello.
Yes.
I'm audible, sir.
Yes, sure. I can
Uh,
hear you.
Sir, I am from India, Maharashtra,
Yeah,
and I am the student of MSc Zoology.
right.
And I want to take a project of odonets on the river, which is uh, Tapi and Purna.
Okay.
So
Which
this
area in
is,
Maharashtra are you?
sir, I am from the Jalgaon,
Oh, okay. Right.
Jalgaon state. Yes, sir.
Right.
So, sorry, sir, I'm not very good
No,
at
no,
much
no.
English.
That's fine. That's fine. Go ahead. I can understand you.
Okay. So, sir, my father is an ornithologist, and
Right.
in Hatnur, we have a Hatnur Dam near on this
Hmm.
area. So, my father, from last 25 years, my father took so much data of migratory birds, and I want to
Hmm.
take an insect data for the upcoming projects like an IBA and
Right,
important
right,
bird
right.
area.
Yeah, yeah,
Yes.
yeah, 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 yeah.
Uh, so we want much bigger status for the Hatnur Dam.
Right.
So how can I collect data and
Yeah. In
what
general,
is
I
the
would say general in any survey, any field survey, your question and this is applicable for others as well if you are planning any service.
yes
If you are doing any dragonfly service, you first you what you have to do is identify the aquatic habitats in your study area, whatever the area, whether it's a dam, there could be if it's a dam, there's a downstream river is there, the upstream river is there, the dam area where you have water is there, then there could be small rivulets and other things that joins into the reservoir. So there could be different structurally different aquatic habitats. First identify those habitats, and then you can use any survey method suitable for insects, and it could be just like birds. For birds we do if you are familiar with the survey techniques, we do transect. Something like transect means you have fixed distance or a fixed time, and then you walk along a line, a path, and then record whatever you observe. Or you do some what we call point count. So you stay in a stationary point and then count everything that you can observe around you. So there are different survey techniques. I'm not going to go into the details on all these nitty-gritties on these techniques, but multiple techniques are there. So you can use any of these techniques. Usually, what we usually do for dragonflies are transects, but then there are other cases where people have used point counts as well, small circular points plots. So yeah, that depends on the habitat that you're working on. Um, then you can visually observe the species. And if you are doing a survey like this, I would I would encourage you to do it at least for an year, because there's a huge seasonal pattern, as I mentioned, especially if in a place like what you are referring to, Maharashtra, you definitely have a significant seasonal cycle on certain. Most species are only out there in the rain season, whereas during dry season you won't see anything. So make sure that your survey is at least it covers 12 months if possible. Uh, if it's not like if you're doing an undergraduate project or something, then you won't have 12 months, but then try to cover as much seasonal variation as possible. And then you can use that data to come up with all these biodiversity indexes based on dragonfly diversity. So that's the secondary part, that how to analyze and present your data. But yeah, those are the few key things. Identify the aquatic habitats. Make sure that you do the surveys throughout, if possible, throughout the year, and then use a standard technique to make counts or other observations on individuals. So that's what you do.
So there's a question by Sarah. Why do some dragonfly species mimic males in appearance?
Certain dragonflies mimic males in appearance. There could be various reasons, and they don't actually mimic the males in most cases. Especially like the androchromes that in themselves dragonflies, where I showed earlier, that's not like a active mimicry. But then certain species have been known to uh, mimic males to reduce the pressure from other males for mating, because if they look more like a male, there's a less chance that these individuals are getting harassed by males for mating. Because during mating season, when it comes to mating, when there are hundreds of individuals out there, every male is trying to find a female and grab onto the female and then mate. So that's how their evolution, the process has been, how they have been adapted. So in certain species, it has been documented that females develop male appearances to make sure that the hassle, the number of males that attempt the mating attempts are less, so they have more, they can mate with better males and have more, invest more energy into the mating process. That's one reason for that mimicry. But in certain other species like these one I mentioned earlier, Eurothemis signata, I showed you photographs of that red color dragonfly. That's I don't that's like a very occasion, very rare kind of sighting. That's more like a variation than a mimicry.
Okay, so there's one more question from Saif. That is, dragonflies tombo have long been celebrated in Japan as symbols of courage and victory. How much of this symbolism is rooted in their actual behavior and traits? Kindly guide.
Oh, that's an interesting question. I am not very familiar with the Japanese culture when it comes to dragonflies, but I know that dragonflies they have kind of a uh, prominent place in Japan's culture, but I'm not very familiar with the exact interpretations. But then I would say that one reason behind it could be that dragonflies are like they are the apex predators when it comes to aquatic environments, when it comes to the micro world around aquatic environments, the insect world, that they are the apex predators. So that might be one reason, and they are known for their voracious predatory behaviors, and also there are superior flight abilities than most other animals. So there could be multiple reasons for this, to suggest that they are kind of superior than the other animals. So it transforms into uh, you know uh, the fundamental, that aspect of victory and courage. So that could be one reason. I'm not very familiar with the cultural aspect of this Japanese culture, so I can't give you a specific answer, but then I assume that could be that could be one of the reasons behind that.
Okay, thank you, sir. There is one last question. After
Hmm.
this, we can conclude the session. How to get a project on Odonta for Dudhwa Tiger Reserve, Uttar Pradesh?
Can you repeat that?
Vipin is asking how to get a project on Odonta for Dudhwa Tiger Reserve in Uttar Pradesh.
Okay, if he's referring to how to do a project, then I would say I would uh, refer back to my earlier uh, answer to that Maharashtra dam incident. Same thing. Dudhwa Tiger Reserve is also, I mean, Dudhwa is a very large area and also very ecologically diverse kind of area. You have multiple huge, you know, diversity of habitats available there. So if you are planning any projects for dragonflies in place like that, you definitely have to make sure that you survey each different habitat type that you can may at least major aquatic habitat types, and also do kind of follow the same protocol. Do an annual survey, at least cover the 12 months, at least one cycle of 1 year cycle, and also especially because that area that like lower Himalayas, that places like Dudhwa would have many rare interesting species. So all these forest species are usually very seasonal. So you have to make sure that you cover all these different seasons and use a standard survey technique and continue the survey. So that would provide you very good results in a place like that.
Okay, sir. This is the last question.
Yeah.
Can you mention some authentic websites for identification of ordinates or any books?
Right. The only reliable website usual reliable website that I'm familiar with is the Indian Ordnata website, which has a lot of photographic observations of many different color forms, many different uh, of many species. For many Indian species, there are multiple images of different like males and females, immature ones, different color forms and everything. Uh, I In most cases I found it very reliable. There are mistakes in the website. I'm not saying that it's perfect. There are mistakes. There are misidentified images like that, but generally it's very reliable. Uh, and also, yeah, that's the only website that I'm usual that I know of. But there are other blogs that are done by several individuals, especially when it comes to Southeast Asian region. There's a Thai Odonata blog. There's a Vietnam Odonata blog. There's a if I'm not mistaken, there's one. There's one for Nepal. I think there's one for Bangladesh as well. So there are multiple blogs. Uh, I've never used them. I've used them, but then I mean I've gone through their material, but I've never used it in a very have not given enough attention to that. So I don't know how there could be certain mistake. There could be certain, but they are very passionate individuals who are running these websites. They are doing great jobs there. So you can see many different forms of dragonflies and different photographs, different morphological forms and everything. So yeah, there are multiple web sources out there. But then on the other hand, when it comes to blogs, they are not like identification based websites. They record whatever that they see. It doesn't mean that they record everything that is out there. So there may be gaps of knowledge gap between what is out there and what is available on the website. um iNaturalist in general, even though it is not a identification kind of a platform, still if you go through their observations, you can uh, use that to identify your own observations as well. If you are referring to books and other field guides, there's no single field guide that can cover the entirety of the Indian region, I would say. Uh, there are field guides for Sri Lankan dragonflies. There are field guides for certain Indian states, but very few. Well, none of them cover more than like 100 different species. So if you are interested in learning more about the entire diversity, you would that won't give you a complete picture. But still, there are multiple. There are for multiple different Indian states. There are multiple field guides available. I don't know whether they are available in the market anymore, but that I'm not familiar with. But I know there are books. Yes. Some material are also available online. For an example, that there's a book on Sri Lankan dragonflies, Sri Lanka distribution atlas for Sri Lankan dragonflies, which also has pictures of different species. So generally, common species in the Peninsular Indian region are covered by that book as well. That the PDF, the ebook is available online for free. If you search for that distribution atlas for Sri Lankan dragonflies, you will come across that. So yeah, there are multiple sources like that. So I can't really guarantee that any of these material, most of these material are like 100% accurate. But then when you go through enough material, you learn by yourself, and then you can use that experience to identify your observations.
Thank you so much. There are more questions, but can I request, since it's already been so long, can I request you to share your Gmail or something so that I can reach out to you personally?
Yes, you can find me on Gmail. My email address is apsumanapala@gmail.com. AP for Amila Prasanna Sumanapala. And I'm also available on Facebook. You can find me on Facebook or iNaturalist if you're there. I usually yeah, I'm not there all the time like I used to be, but I time to time I check iNet as well. So if you have any identification uh, observations that you need to get identification, you can drop me the link, iNet link, upload it to iNet, and drop me the link in the chat so I can have a look at it. Yes, so yeah, you can contact me through the general social media. I'm usually available in all the platforms.
The spelling of Sumanapal, am I
S
pronouncing it
U
correctly? Would
A
be
P, yeah, AP for Amila Prasanna, and then Sumanapal is SUMANAPALA. You can
P-A-L-A.
find my name on that post that.
Yes, sir. Okay. Thank you so much for conducting the session, sir.
Sure.
Thank you so much for sharing this knowledge with us. Uh, and thank you for the participants for joining.
Yes, thank you everyone. Yes, have a good night.
I'll just share your mail ID and then I'll stop the meet.
Sure. Okay. Thank you.
Thank you. apsumanapala@gmail.com, right? Thank you everyone for joining.